Hello everyone. So in today's session we'll be discussing the video solution of the question that has appeared in GATE Life Sciences question paper. The topic which we have chosen for today is from enzyme kinetics and inhibitors. So this is our question, which is question number 19 from biochemistry portion from GATE Life Sciences 2012 question paper. So this uh, question states that the kinetics of an enzyme in the presence or absence of a reversible inhibitor is described in the following graph. So, if the concentration of reversible inhibitor in plus I experiment was equal to 3 into 10 to the power minus 3 molar, then the dissociation constant for the enzyme inhibitor complex is. Okay, so let's start from the basics. In the question, it is stating reversible inhibitor. Okay, so in an enzyme kinetics, we have chemicals that can affect the chemical reaction. Those are called as we can say negatively affect, so those are called as inhibitors. And in that also we have two categories. One is the reversible inhibitor category, another is the irreversible inhibitor category. Okay, so in irreversible inhibitor, so this irreversible inhibitor permanently damages the functional group of enzyme. No, does the permanent damage to the functional group of enzyme or the active site of the enzyme. That is why the enzyme remains non-functional. It becomes non-functional and it doesn't no chemical reaction would occur in case of irreversible inhibitors. Okay, so one example of a reversible inhibitor is cyanide. So as we know, cyanide can bind to the enzyme. It is an irreversible inhibitor. It can bind to the complex 4 of mitochondrial ETC, which is cytochrome oxidase enzyme, which is involved in mitochondrial ETC. In this mitochondrial ETC, its purpose is to produce ATP. So if cyanide is affecting cytochrome oxidase, it is hampering the mitochondrial ETC, and when the mitochondrial ETC will hamper, the ATP production will stop. And as we know, all our activities are dependent on the ATP production. All the physiological processes in our body, all the physiological processes of our body will also be hampered. That is why cyanide is a very toxic chemical for our body, because it works as an irreversible inhibitor for cytochrome oxidase enzyme. Another example of this irreversible inhibitor is one inhibitor called DEF. Okay, so this is the structure of that inhibitor. This is DPF, and one enzyme which has the active site. This is the active site. So this fluorine attack to the lone pair of the oxygen and bind and make the structure. So now this is the functional group of the enzyme, which is CH2OH, which is now lost after binding with this DPF inhibitor. So this is a permanent damage to the active site of the enzyme and leaving this enzyme non-functional. So this is the acetylcholine esterase enzyme. Common in DPF generally inhibit uh, acetylcholine esterase enzyme. So DPF inhibiting this enzyme by this mechanism making it its functional group non-functional. Another example of irreversible inhibition is suicidal inhibitors, which is also called mechanism based inhibitor. So these utilize the enzyme substrate kinetics mechanism, make a more stronger enzyme inhibitor complex which cannot be reversed back and cannot be recovered, and which causes ultimately suicidal inhibition for the enzyme's function. So for one example of suicidal inhibitor is allopurinol. Okay, it's an example. So these are our irreversible inhibitor which causes a permanent effect on the enzyme and cannot be revert back. Okay. On the other hand, we have reversible inhibitor which effect can be reverted back. So in reversible inhibitor, we have three categories: competitive inhibitor, competitive inhibitor, uncompetitive inhibitor, and non-competitive inhibitor. Okay. So in competitive inhibition, we have an inhibitor which competes for the active site of enzyme. So we know that enzyme have an active site where the substrate will go and bind. But the inhibitor, which is also similar in structure of the substrate, structurally similar to the substrate, this can also, because it is structurally similar, this can also go and bind to the active site of the enzyme. So now that there is a competition between substrate and inhibitor to bind to the enzyme structure. So because of this competition, the affinity of substrate in presence of inhibitor uh, decreases. So because of the decrease in the affinity, our Km value decreases in competitive inhibition, but the Vmax value remains the same. No change in the Vmax value in competitive inhibition. Okay. Now we have another category which is uncompetitive inhibitor. So uncompetitive inhibitors, what they where they bind. So as this inhibitor was binding to the free enzyme, but the uncompetitive inhibitor will only bind to an enzyme which is already bound to a substrate. Okay. So it already binds to enzyme substrate complex, and as it is binding to the enzyme substrate complex, it is reducing the Vmax as well as the Km. So Km will increase because here in this case Km will increase because more the Km, lower the affinity. Here the affinity of enzyme with substrate gets increased because the inhibitor will work only on the substrate bound enzymes. So here the Km as well as Vmax, both the values get decreased. Okay, but in non-competitive inhibition, what is happening? It is binding to the inhibitor. Okay, so this is my, okay, it can bind to the only to the free substrate. Okay, so we have generally like one active site, and other than active site we have another site which we call as allosteric site. So inhibitor will bind to the free enzyme only, and that binds to this allosteric sites, the inhibitor. So as it is binding not to the active site, that is why in case of non-competitive inhibition, there is no change in the Km value, but it affects the reaching the maximum velocity. It decreases the maximum velocity of the reaction. So these are the kinetics parameters in case of competitive, uncompetitive and non-competitive inhibitor, and what are their mechanism of action. So competitive inhibitor can only bind to the free enzyme, and it increases the Km, and Vmax remains unchanged. Uncompetitive inhibitor could only bind to the enzyme substrate complex, and it decreases both Km as well as the Vmax. Non-competitive, okay, yeah, so it non-competitive inhibitor can bind to free enzyme as well as the enzyme substrate complex, and it does not, as it is binding to the allosteric side, it does not affect the Km, but it definitely decreases the Vmax of the reaction. Okay, so if we'll see the graph, the Lineweaver-Burke plots for the enzymatic inhibition will give three different. So all these blue graphs are showing us without any inhibitor. So here, competitive inhibitor, we are saying that it is uh, increasing the Km value, that means decreasing the affinity of substrate towards the enzyme, but Vmax remains the unchanged. So here you can see in the Lineweaver plot, we have at the y-axis, the intercept is called as 1 by Vmax, and the x-axis, the intercept is called as the 1 by Km. This is in presence of inhibitor, is 1 by Km. Okay, so if we are uh, showing the change with an alpha factor, so here in this case, the new Km is increased by an alpha factor, but the new Vmax doesn't change, it remains the same. Here, the new Km is decreased by alpha, and as well as the Vmax also decreased by alpha. In the non-competitive one, where the Km is not being affected, new Km and old Km are same, but Vmax is getting decreased by alpha. Okay, so let's try to solve the numerical based on this information. So here, the kinetics of an enzyme in the presence or absence of reversible inhibitor. So it is talking about a reversible inhibitor, either of three: competitive, non-competitive, uncompetitive. So here, seeing the graph, you should know what is the which reversible inhibition is taking place here. So here we can say the y-intercept is same. That means the Vmax is not changed. But in our, so out of three, in all, in these two, Vmax is being changed, but not in this one. And also the Km value is increasing. The new Km value is increased. So here if we'll see plus inhibitor, we are getting higher value. We are getting a higher value that says that the Km is increased. So that is why we can find out that this is a competitive inhibition example. Okay. So the question is being asked that if the concentration of reversible inhibitor in plus I experiment was equal to 3 into 10 to the power minus 3 molar, then the dissociation constant for the enzyme inhibitor complex. Okay, so when enzyme and inhibitor bind, make, so this is the dissociation constant is calculated for this particular reaction. Okay, so to calculate, so this is the formula to calculate the dissociation constant. Alpha is the alpha value, the factor with which we are seeing the decrease and increase. I is the inhibitor concentration, Ki is the dissociation constant which we are supposed to find out. Okay, so here as we have found it is a comparative inhibition, Vmax remains the same. So now we have to find the alpha value, we have to get the alpha value. As we know the Km is being changed, so the new Km is alpha upon old Km. So here the value which we are getting here is in the form of Km, 1 by reciprocal of Km. Okay, so 1 by Km is equal to minus 4. 1 by Km. As we know that substrate value cannot be in minus, so we'll reduce the, ignore the minus, and these are our Km and Km prime values. Okay. So, alpha Km. So if we put these values, so alpha value will come 2. Now this value we will be keeping here. Inhibitor concentration is given to us 3 into 10 to the power. Again, lost it, sorry. So this, oh god, sorry. Undo. It's okay. So I hope you got it. So alpha value we are getting 2. So this alpha value we will be putting here. So this will be 2. And the enzyme concentration given to us 10 into 10 to the power minus 3 molar. Okay. There will be 3 into 10 to the power minus. This will be our dissociation constant. So if the option will see, C option is the correct option. So dissociation constant for enzyme inhibitor complex is this. Okay, this would be the correct option. So similarly, you need to remember this particular formula, and also in case of any enzymatic conversion, to get the alpha value, you need to remember all the graphs, 3 graphs, and how their values are changed, and where how to find out the values of alpha. Okay, seeing the values. Okay, this is my NPTEL reference. Thank you for listening.